Hi everyone. Chapter 6, Expression of Biological Information. For 6.2, at the end of this video, students should be able to explain the DNA replication. Let's watch this video to give an overview of DNA replication.
DNA. We talk about it so much, it's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when? When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3', and the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one.
There are 7 enzymes and protein that involve in DNA replication. Number 1 is helicase. Number 2 is topoisomerase. Number 3 is primase. Number 4 is DNA polymerase 1. Number 5 is DNA polymerase 3. Number 6 is DNA ligase. Number 7 is single-strand binding protein or SSBP. Now, let us explore the process of DNA replication and the function of these enzyme and protein. Replication fork formation. Before DNA can be replicated, the double-stranded molecule must be unzipped into 2 single strands. In order to unwind DNA, these interactions between base pairs must be broken. This is performed by an enzyme known as DNA helicase. DNA helicase disrupts the hydrogen bonding between base pairs. This area will be the template for replication to begin. After the parental strands separate, single-strand binding proteins bind to the unpaired DNA strands, keeping them from repairing. The untwisting of the double helix causes tighter twisting and strain ahead of the replication fork. Topoisomerase helps to prevent supercoiling due to the tension that created during unwinding of DNA strand. Primer binding. Once the DNA strands have been separated, a short piece of RNA called a primer binds to the 3' end of the strand. Primers catalyze the synthesis of RNA primer. The completed primer, generally 5 to 10 nucleotides long. The new DNA strand will start from 3' end of the RNA primer. Elongation. DNA polymerase 3 binds to the 3' end of the RNA primer and begins adding new base pairs complementary to the RNA primer. Then, continues adding DNA nucleotides that complementary to the parental DNA strand template strand. The replication fork is bidirectional. One strand is oriented in the 3' to 5' direction of leading strand while the other is oriented 5' to 3' of lagging strand. For the leading strand, The newly formed strand is continuously toward the replication fork. While for lagging strand, replication of new strand occurs away from the replication fork. The lagging strand begins replication by binding with multiple primers. Each primer is only several bases apart. DNA polymerase III then adds pieces of DNA nucleotides to the 3' end of the RNA primer, causing formation of several pieces of fragment called Okazaki fragments. After Okazaki fragment forms, DNA polymerase I replaces the RNA nucleotides of the adjacent primer with DNA nucleotides. Termination. Once both the continuous and discontinuous strands are formed, an enzyme called exonuclease removes all RNA primers from the original strands. These primers are then replaced with appropriate bases. DNA ligase catalyze the joining of Okazaki fragments together, forming a single unified strand. Once completed, the parent strand and its complementary DNA strand coils into the familiar double helix shape. In the end, replication produces 2 DNA molecules, each with one strand from the parent molecule and one new strand. So, you have homework to do. Please define each of the terms stated and submit to your tutorial lecturer. Thank you.